Good morning dear students, myself Lavanya. I am going to take anatomy in flowering plants today. Okay, first, what is meant by anatomy? Anatomy is study of internal structure of plants, that is plant tissues. We are going to study detailedly about whatever the tissues are there in the plant and what are their functions, how the cells are arranged, all that we are going to see. Right, anatomy, first each and every organ and organism, systems, everything is going to be started through cell. Right, these cells will get organized to form tissue, then to tissue system, then tissue systems are going to get organized and forms organ, then to organ system, then finally to organism, that is plants. Right, in all these, what we are going to study in this chapter as a gist I am going to mention here itself. Look at here, under tissues we would be discussing about meristematic tissue, and then permanent tissues. Under tissue system, vascular tissue system, ground tissue system, epidermal tissue system. Right, under this we are going to discuss this. And under organ, all the organs starting from root, then stem, leaf, flower, fruit, seed. This we are going to discuss. Organ system, root system, shoot system. Okay, in this chapter as a whole, all these we are going to discuss in detail here. Okay, next let us start first with meristematic tissue. Meristose means thread-like structure. Meristose, dividing cells. Okay, dividing cells. These are dividing cells. And these cells are thin, having dense cytoplasm. Dense cytoplasm. Due to presence of meristematic cells only, cells are going to show the growth, repair. Okay, damaged cells are going to be replaced by the new cells through meristematic cells only. And these meristematic cells are not present in the entire parts of the plant. And based on the location, and based on the nature, it is divided into primary meristems and secondary meristems. Under primary meristems, these are seen from beginning of the plants. When the life of a plant started, when origination of a plant happened, at that time onwards we can see these cells and these tissues. At the beginning, these are found from the beginning. And if you take, just for our reference only I am drawing this picture, imaginary picture. Look at here, this is the shoot apex and this is the root apex. Okay, so this is root apex, tip, this is shoot apex. And when the sun, based on the availability of a sunlight, hormones are going to reach to here, and those hormones are going to activate the actively metabolically actively cells called as meristematic cells. Those cells only, primary meristematic cells, they get activated and they show growth in the lengthwise in the shoots, that is stem. And you come to the root, here also you have cells, meristematic cells, and you may get a doubt that here sunlight is not available, how those cells are going to get activated. Yes, here moisture and the minerals whatever present is going to make them activated, and then their growth is going to happen in search of water that is in the moisture content, and the roots are going to develop towards the geotropic movement. This is phototropic movement. So we can see the increase of a length increase in the stem and also the root. That is possible by the meristems, primary meristems. These meristems which are present at shoot tip and root tip is named as apical meristems. Apical meristems. Right, and next you see here we can see at the nodes, internodes and nodes, there also we can see the growth, width increase and elongation is happening. That is because of a tissue present, intercalary meristems. These meristems are called intercalary meristems. See, cattle like buffalo, goat and cow and all, when they feed on grass, okay, grass or any plant, after eating they won't eat completely, when they leave some part of it, again that leaf part or the stem part is going to grow again. What is making them to grow again? Because of presence of intercalary meristems. Because of these intercalary meristems only growth is happening effectively. Okay, this is the part related to primary meristems. Next, let us see about secondary meristems. Secondary meristems are formed from permanent tissues. They are formed from permanent tissues. They are formed from permanent tissues. Now, see, based on the location and the need, because this is going to bring the secondary growth, help in the secondary growth of a plant. So, based on the location, this is divided into types. Cork cambium and vascular cambium. Okay, this one again, fascicular cambium and interfascicular cambium. Here and all we can see the development. Growth is, it is going to show the growth. Okay, this is about primary and secondary meristems. With this we have completed meristems. Next, moving to permanent tissues. Permanent tissues. These are the tissues which are formed from meristematic cells only. See, meristematic cells which are metabolically active, they are going to get differentiated into permanent tissues. It doesn't mean that it has lost completely its cell division ability, but it cannot divide as it is like a meristematic cell. Okay, that makes the difference here. Permanent tissue cells are cells are from meristematic cells only, from the meristematic cells which undergoes differentiation, means getting organized to perform different functions and forming some structural unit, that is simple permanent tissues. Simple permanent tissue and complex permanent tissues. First here, simple permanent tissues, look at here. Simple, the name itself is going to give an idea that the simple organization of a cells can be seen. Mostly single type of cells. Single type of cells are found. Cells are found. That means you remember their function also is going to be the simple, because if one type of cells are there means they perform only one kind of a function. When there is a multiple or many cells, different types of cells are there, then they can perform different functions, right? So under simple permanent tissues we have three types of cells. First, parenchyma, collenchyma, and then sclerenchyma. Now let us see one by one. Parenchyma, collenchyma, sclerenchyma, right? First one is parenchyma. See, here the parenchyma is a living cell. It is a living cell. Even collenchyma also living cell. But whereas when you are going to come sclerenchyma, it is a dead cell. It is made up of dead cell. Dead cell means, is it going to have a nucleus, that means protoplasm and all? No, it is not going to have all those things. Okay, cell that is just present. Cell that is just present. Right. Now this parenchyma cells are isodiametric in shape, or they can be polygonal in shape. Okay, etc. Right, here you can see collenchyma cells can be oval, polygonal, or round. Okay, based on the function they perform and the location, their cell shape is going to get organized. Whereas coming to sclerenchyma, it can occur in two forms. It can occur in two forms. One is fibers and other is scleroids. Okay, if you see fibers, its arrangement is like this. Like this, and it is surrounded by thick cell wall. Okay, when you come to scleroids, broad lumen can be seen and thick cells are surrounded around him, around that cells. Come back. Coming to parenchyma, the cells can be of isodiametric or polygonal, etc. And cells are continuously arranged, no intercellular spaces. If present, also very negligible. Here also cells are compactly arranged without any intercellular spaces. Arranged. And coming to parenchyma, they have the chloroplast. So they can perform photosynthesis. One of the function of parenchyma is to perform photosynthesis because they are provided with chloroplast. And collenchyma also has chloroplast, that's why it can perform photosynthesis. In addition to that, it is going to help in providing mechanical support. providing mechanical support. That is one thing we can say. And how to identify just by seeing these cells among these three means it is very easy to identify sclerenchyma because they can be either fiber or a scleroid. But whereas when you come to collenchyma, you can identify them those cells at the corners. Okay, you can see the corners and all having depositions, thick corners. They have thickened corners. They have thickened corners due to cellulose, hemicellulose and pectin deposition. Cellulose, hemicellulose and pectin deposition. Okay, right? Whereas those cells you can identify easily. And apart from this, if you see sclerenchyma, okay, sclerenchyma has lignified walls. Lignified walls, usually those lignified walls are useful in forming a secondary cell wall. They have lignified walls, cell wall, right? And they are going to form pulp of sapota, guava, right, pear. Okay, all this is going to form. Even this is found in nuts also. We can see them in the nuts also. And leaves of tea. In tea leaves also we can found this. Okay, and several in seeds of legumes also. Okay, this is about completely simple tissues, simple permanent tissues. Hope you understood this very well, right? And next let us go for complex permanent tissues.